Hi everyone. This video is about the important key concept when we answer the questions that's related to the link gene. Questions on the genetic can be asking on the link gene, and it can be asking about the link gene as well. So that's why it is important for us to differentiate the questions that is related to unlinked gene or the linked gene. And at the same time, we need to know how to write the genotype of the parent and also the offspring as well. So that's why for the first concept, we need to differentiate. We need to know how to differentiate the unlinked gene and also the linked gene. So basically, unlinked gene is those pair of gene that occurs on different chromosome. Yeah, for example, this pair of gene is occurring on one homologous chromosome pair, and B gene is on another chromosome. They are not related. So this type, they are considered as the unlinked gene. This will include all those normal Mendel's model, the dihybrid cross, or even the deviation from the Mendel's model, like the codominant, incomplete dominant, polygenic inheritance, multiple alleles, all of this, it is mostly unlinked gene. While for linked gene, it is basically the genes that occurs on the same chromosome. Yeah, the gene that occurs on the same chromosome. And based on the distance of these two genes on the chromosome, we can further divide them into two. The linked gene, it can be physically linked but genetically not linked, or it can be physically linked but genetically linked as well. For those genes that's physically linked but genetically not linked, that's basically those genes that occurs on the same chromosome. That's why they are physically linked, but they are too far away. Yeah, they are too far away, so that's why they are genetically not linked. While for those genes that occurs on the same chromosome but very close to each other, so that is considered as they are physically linked because they are on the same chromosome, but genetically they are linked as well because they are too close to each other. The way to write the linked gene, no matter is the genetically not linked or genetically linked, is the same basically. It will be the same. Those two genes that occurs on the same chromosome, we will write it together. And then this small a, small b that occurs on another chromosome, well, we'll write it together. Same thing goes to those linked gene that is genetically linked. So in the questions of the linked gene, right? So sometimes it is quite confusing, yeah, when we want to differentiate whether the big A, big B should be on the same chromosome or the big A, small b should be on the same chromosome. Because sometimes we are not sure whether it should be written in this way or this way. Yeah, we don't know whether we should write like this or write like this. So yeah, this is especially true when we need to identify the genotype of the parents. So how we can easily differentiate them, how we can easily find out or assume that the genotype of the parent should be this type or should be this type. So actually it's quite simple. We can look at one of the example here, which is this one. So as you can see here, the round and the smooth, yeah, round and smooth are both dominant. Yeah, these two are dominant trait. Okay, while the oval and the rough would be the recessive trait. Yeah, we look at this. So this would be the important key for us to identify the genotype of the linked gene in parents. We will look at the ratio. For example, for this one, this is the number of the offspring, right? So this, after we calculate it, we will get this ratio. So we will look at those bigger ratio and those smaller ratio. If you find that the offspring, with both dominant trait, you see that? Round, smooth, both dominant, and also both recessive, oval, rough, both also recessive. We will look at this combination, yeah? So those offspring with both dominant and both recessive, if let's say their ratio is smaller, if their ratio is smaller, then that means that it will be this type. The parent will have this type of the genotype. One of the parent must have this type of the genotype, this type of heterozygous. That means that one dominant and one recessive on the same chromosome, and then one recessive, one dominant on another chromosome. On the other hand, if you find out that those offspring with both dominant and those offspring with both recessive, they are having higher ratio. If let's say this one is 12, and then these two, it become 1 and 1. Okay, so if you find out that these two, both dominant trait, both recessive trait, this type of the offspring, they are having higher ratio, then that means that the parent, one of the parent, the heterozygous parent, must have this type of the genotype. That means that both dominant allele should be on the same chromosome, both recessive alleles should be on the another chromosome. So this is basically how we can easily differentiate that. Yeah. So to repeat that, if let's say it's like this, if the parental genotype, the heterozygous parent, yeah, the genotype is in this way, both dominant on one chromosome, both recessive on another chromosome, then that means that yes, those offspring with both dominant trait and both recessive trait will have higher ratio. And if let's say the parent is having this genotype, that means that one big A, one small b here, then one small a, one big B here. If let's say it is different, it is this type of genotype, then that means that the offspring, the offspring with both dominant trait and both recessive trait will have lower ratio. So that is actually the key for us to differentiate when you face the questions that's related to linkage and asking you to find the genotype of the parents. So that is the first part of the concept. Then next, we'll move on to the concept number 2. So concept number 2, well, when we do the questions that's related to the linkage, normally we will look at the ratio of the offspring. And if let's say it is unlinked, if let's say it's unlinked, okay, so normally, yep, we will do the test cross, and then we will find out that the number of the parental and recombinant offspring, the parental offspring, recombinant offspring is almost similar. Yeah, it's almost similar. Well, if let's say it's a linked gene, but it's genetically not linked, that means they are far apart, right? When you do the cross, you do the test cross, you will find out that the parental and the recombinant, same thing. Their number of the parental and number of the recombinant offspring is also almost similar. The only one that will give you too high ratio for parental and then too low ratio for the recombinant would be those that is genetically linked. Those gene, those pair of the linked gene that is close together, on the same chromosome, only that, yes, when you do the test cross, you will see that there will be much more parental and then much lesser recombinant. So in other words, when you see the ratio, when you see the ratio, the test cross, the parental and recombinant, right? Because the number of the offspring is almost the same. So they will give you the ratio around 1 to 1 to 1 to 1. Same thing goes to the genetically unlinked one. Same thing goes to this type, linked gene, but they are too far apart. So they will also give you almost the same ratio, 1 to 1 to 1 to 1. Only, only this one, only those that is linked and they are occurring on very close on each other, very close with each other on the same chromosome, they will give you a big ratio for parental, small ratio for recombinant. So that's when it gives you, like, for example, it can be 4 to 4 to 1 to 1, it can be 10 to 10 to 1 to 1, it can be 9 to 9 to 1 to 1, or 12 to 12 to 1 to 1, just like this question. 12 to 12 to 1 to 1. So that's why, this one, it will be two parental, will be huge. It can be, let's say, I just simply put, I will just put 9 to 9 to 1 to 1, let's say. So you can see that the recombinant will be much lesser. And when we do the questions, yeah, please remember that in order to achieve this ratio, in order to get this type of ratio, no matter it is for the unlinked gene, or it is for the linked gene but genetically not linked, or it is for the linked gene and genetically linked gene. Yeah. In order to get this type of ratio, it must be a test cross. It must be a test cross. Yeah. So please remember that. Okay. And normally to get this ratio, it would be heterozygous test cross. Yeah, it will be a heterozygous test cross. So that means that it must be a heterozygous cross with the homozygous recessive. Same thing here. Heterozygous cross with homozygous recessive, heterozygous cross with homozygous recessive. When we doing this type of test cross, then only then it is possible to get this type of ratio. If you are not doing the test cross, then you won't get this type of ratio. Yeah, so that's why this is another thing that can help us to solve the questions and then prevent the confusion. So this is the second concept. Then next we'll go for the third concept. So for the third concept, what is the reason why we will have this type of ratio when we do the test cross? And what is the reason of having this ratio? Okay, so please remember here, as you can see this, for unlinked gene, yeah, for unlinked gene, well, these will be the parental. If let's say these two go together, then they will be the parental. These two go together, it will be the parental. And if let's say this A and this B go together, this A and this B go together, then it will be the recombinant. Okay, so why we will have the parent number of parental and number of recombinant almost similar? Well, the reason is because the production, how the recombinant type offspring can be produced, right, is because of the independent assortment. It's all due to the independent assortment. Because when we want to produce a gamete, the homologous chromosome will be separated during the meiosis one, during the anaphase one. Homologous chromosome must be separated. They must be separated. This is 100% will occur, right? It will 100% it will occur. So that's why for independent assortment, it is something that must occur. So it can be random. It can be these two homologous chromosomes go together. It can be these two chromosomes go together. Or it can be this one, this chromosome and this chromosome go together, and this chromosome and this chromosome go together. So it's totally random, totally random. That's why it's called independent assortment. And because of this, the chance of producing the recombinant is similar to the chance of producing the parental. Yeah, so because of this, yeah, the chance that due to the independent assortment, the chance of producing the recombinant gamete or then after that recombinant offspring is similar with the chance of producing the parental gamete and after that parental offspring is the same. It is similar. So because of this, they have same chance. So that's why they will have the same ratio. The ratio become 1 to 1 to 1 to 1. The parental and the recombinant is the same. Well, it comes to this one. When it is a linked gene, but they are very far apart on the same gene. Yeah, so it's on the same chromosome. They are very far apart. So for this type, what is the reason? What is the reason why they will have this 1 to 1 to 1 to 1 ratio? Why the recombinant will be having the similar chance as the parental? Why the number of the recombinant offspring will be same like the parental offspring? So this one, the reason is due to crossing over. It's due to crossing over. So same thing, when crossing over occurs, it will exchange. If not, it's almost impossible, right? Because the big A and big B will tend to go into the same gamete, and small a, small b will go to the same gamete. So during the meiosis one prophase one, crossing over will happen. It must happen, right? Crossing over must happen. So the thing is, because the distance is too far away, yeah, there's big distance in between these two genes. So that's why the chances of crossing over and exchange this gene, exchange this allele, will be very high. So since the chance is so high, so it is causing the chance to produce the recombinant gamete similar to the chance to produce the parental gamete. And because the chance of producing both type of gamete is the same, so that's why the chance to produce the recombinant offspring is similar to the chance of producing the parental offspring. So that is the reason. Yeah, that's the reason how we explain that. Well, lastly, for this one, for those genes that is very close to each other, yeah, close to each other on the same chromosome, this type of linked gene, they will normally give you this type of ratio, much more parental and then much lesser recombinant. So why the the number of the recombinant offspring is so low compared to the parental? Again, it is due to crossing over. Yeah, again, it's due to crossing over. Okay, so crossing over, yes, it will occur during the meiosis one, during the prophase one. It must occur, but the thing is, the chances is very low because they are too close to each other. You can see the A gene and B gene, they are too close to each other. So crossing over when it occurs, let's say the crossing over occurs here, then it won't break the big A and big B. It won't break the small a, small b. The big A, big B will still go into the same gamete. The small a, small b will still go into the same gamete. So they won't, the chances for crossing over to occur and then exchange them will be much lower, much lower. Okay, because crossing over can occur here, then nothing happened. It won't change. The crossing over can occur here, then nothing change. Okay, yeah. So that's why the chances, because the distance is low and the distance is very short, so that's why the chances for the crossing over to occur and exchange the gene will be low as well. So that's why the chances to produce the recombinant gamete is much lower compared to the chance to produce the parental gamete. And because of this, that's why the chances to produce the recombinant offspring will be lower, and the chances to produce the parental offspring will be higher. So that would be the third concept. Then next, we'll move on to the concept number 4. So for concept number 4, well, same thing again, right? So this part is all on the linked gene, unlinked gene. This part is all on the link, physically linked but genetically not linked. This one will be the physically linked, genetically linked. Concept number 4 is on the recombinant frequency. When we count the recombinant frequency, as you can see the ratio just now, yeah. So the unlinked gene, they will give us 1 to 1 to 1 to 1. So you can see that the ratio of the recombinant is almost equals to the ratio of the parental. So because of this, when we calculate the recombinant frequency, we take the recombinant divide by total, then times 100%. When we calculate the recombinant frequency, we will find out that the recombinant frequency is almost 50%. Yeah, the reason is because the number of the parental and the number of the recombinant offspring is almost the same. So that's why we will get almost 50%. Yeah, of course, because when we look at the population maybe the parent, maybe they will produce let's say 50 children, yeah, 50 offsprings, right? So, well, of course, there will be some fluctuation in the number, yeah. So that's why not necessarily it will get exactly 50%, not necessary, yeah, because it can be the parental, two type of parental, two type of recombinant, right? So maybe it can be producing, let's say, this one may be producing 50, this one may be producing 48, this one may be producing 52, let's say. This one may be producing let's say 50. So you can see that, well, the when you calculate the recombinant frequency, yeah, you take these two, okay, then you divide by the total number and times 100%, it will be 50 point something, maybe 50.1, 50.2. It can be 49.8, it can be 49.6. So due to the number of the offspring that's being produced is lower. So that's why, well, there might be some fluctuation. So it is okay. It is sometimes it will be almost, near very close to 50%, maybe 49.5 and above until 50.5 and below. Yeah, so around that. So that would means that it is actually having the same number of parental and recombinant. So that's why recombinant frequency will be almost 50%. So it will be same for those genetically not linked one. Those linked gene, but genetically not linked one, they will still give you almost the same recombinant frequency. Only those that is physically linked and genetically linked gene, they will give you the recombinant frequency that is lesser than 50%. Yeah, why? Well, the reason is because the parental offspring will be larger number and the recombinant offspring will have smaller number. So that's why, well, they will have the recombinant frequency that's lesser than 50%. So this is when it comes to the questions that's related to the recombinant frequency. And sometimes the questions will ask about the map distance or map unit, ask you about the map unit. Okay, so to calculate the map distance, of course, it depends on the recombinant frequency. It depends on recombinant frequency. Yeah, so you will have to calculate the recombinant frequency. Then only then you can get the map unit or the map distance. All right. So please remember, for those unlinked gene, they don't have any map distance because they are not on the same chromosome. They are on different chromosomes. So when they are on different chromosomes, we cannot count their distance between the two genes. So that's why there's no map distance, even though you will see the recombinant frequency is almost 50%, but then they don't have the map distance. Okay, they don't have the map distance. While for those genetically not linked gene, right? So those linked gene, right, but genetically not linked, they are too far away. Okay, so for this type, although their recombinant frequency is almost 50% or 50%, well, when we calculate their map distance will be more than 50 map unit. It will be more than 50 map unit. Okay, so, well, the reason is because they are too far away already. Yeah, they are too far away. So when they are too far away, so the chances to produce a recombinant will be equals to the parental. It won't be more than the parental. So because of this, the recombinant frequency will exactly give you 50% only. But that does not mean that their map distance is equals to 50 map unit. So this one, you have to be alert on this. Okay, so how to find the map distance for this type of gene, for this type of the linked gene? Well, this one we need to find a relative point. So what it means by relative point? So that means that you have to look into the questions. Okay, so if let's say they tell you that, okay, the gene A and gene B, yeah, gene A and gene B, yeah. Okay, so if let's say they tell you that the recombinant frequency equals to 50%, let's say, or 49.9%, or 50.1%, let's say. Yeah. So when they tell you that, okay, this is the recombinant frequency between the A and B, then what is the map distance between the gene A and B? You cannot directly write 50. Yeah. So you have to be alert for this one because they are too far away. So you need to find a relative point. You need to see whether the questions tell you, for example, gene A and C. For example, if let's say they tell you that for gene A and C, the the recombinant frequency is equals to, let's say, 30%. And then another point is from C to B, the gene C to B, let's say the recombinant frequency is equals to, let's say, another, 40%, let's say. Okay, so that will mean that C would be our relative point. It will be our reference point. So since we know that from A to C is 30% and from C to B is 40%, so can we find the map unit? Yes, the map unit will be equals to, you take this recombinant frequency plus this recombinant frequency. So with that, what is the map distance between the gene A and B? Gene A and B, the map distance will be 30 plus 40. It will be 70 then. Yeah, it will be 70. So that will mean that the map unit will be the map or map distance, yeah, of A to B, yep, it will be 30 plus 40, and the map distance will be 70 map unit. This one, the unit cannot be percentage ADF because percentage is for recombinant frequency. So this must be map unit, 70 map unit. So that is the way how we find the map distance between the two genes that is too far away. The gene, the link gene that is physically linked but genetically unlinked. So this is how we work on this. So how to differentiate between this question and this type of question? So for those type of question that is asking for unlinked gene, well, normally in the questions, we won't tell anything about the link gene. And another thing, we won't provide any reference point. We won't tell you there's the distance between the A and C or the C and B. So this type is considered as the unlinked gene. Well, for those questions that's related to linked gene but too far away, well, normally the questions will tell you the reference point. They will tell you there's a relative point. For example, the gene A and gene C, they are having the recombinant frequency or they are on the same chromosome. Then gene C and gene B is on the same chromosome. So we will tell you, the questions will tell you this, though you have to be alert. While for the last one, which is the physically linked but genetically and genetically linked gene, yeah, those linked gene that is close together. So for this type of the questions, which is quite common, yeah, this is considered as a very common questions. So this type of common questions, when we ask the linked gene that is close to each other on the same chromosome, then basically the map distance will be lesser than 50 map unit. So for this one, when you count the recombinant frequency, it will be lesser than 50% or so. The recombinant frequency will be lesser than 50%. So that's why we can directly take the recombinant frequency, change the percentage to become the map unit. So if let's say when you calculate, it is 40%, then that means that the map distance will be 40 map unit. So that is quite simple. Directly, you can take the recombinant frequency and change it to become the map distance. So this is for those that is linked gene and very close to each other. For those that having lesser than 50% recombinant frequency. So this is the concept number 5 on the map distance. And lastly, for concept number 6, which is the last concept. So for this one, crossover and the recombinant frequency. So it's a general relationship in between that. So crossover and recombinant frequency, well, they are related because of the distance between the two genes. Yeah, because of the distance between the two genes. So generally, what you need to know is that the distance between the two genes is important. Yeah, the distance between the two genes is important. If the distance is very close to each other, the distance is too short, then that means that the crossover, yep, we look at the distance, yeah. And we look at the crossover. And then we look at the recombinant frequency. If the two genes, they are too close to each other, so that means that the distance is very short. If the distance is very short, then that means that the chances of crossover to separate the allele, yeah. For example, this one, the big A and big B allele, well, they will tend to go into the same gamete. Small a and small b will tend to go into the same gamete. Yeah. So if the distance is too short, then that means that the crossover to separate the big A and big B, to separate the small a and small b, the chances of crossing over to produce big A and small b, big B and small a will be lower. So the chance of crossover will be lower. So when the chance of crossover to separate, I should say the chance of crossover to separate, to separate the big A, big B, or the small a, small b will be lower. Okay, not just crossover only, because crossover, it can occur here, it can occur here, it can occur here. Okay, crossover can occur anywhere on between the homologous chromosome. Okay, so this is the chance of crossover to separate the allele. Yeah, to separate the allele. In this case, would be separate the big A and big B and separate the small a, small b to produce a recombinant. Okay, to produce the recombinant. So this one, yep, it will be the chance of the crossover to produce recombinant gamete. So in other words, to produce the recombinant offspring later, yeah. So the chance of crossover to produce a recombinant type will be lower, much lower. And because of this, there will be much lesser recombinant offspring. So because of this, the RF value, the recombinant frequency will be lower as well. So this will refract it directly. The recombinant frequency lower means that the distance is lower. Okay, so when you compare here, when the distance increase, when the distance between the two gene is higher, the distance between the two gene is higher, then that means that the chance of crossover to produce the recombinant will be higher as well. Because if the crossover occurs here, it will produce a recombinant. If the crossover occurs here, it will also produce a recombinant. Compared to this one, it must occur here, then only it can produce the recombinant. So you can see the chances is lower. So this one, the distance higher, the distance is longer, then that means that the chance of crossover to produce a recombinant gamete will be increasing. Yeah, it will be higher. So because higher chance to produce recombinant gamete, so that means that there are higher number of the recombinant type offspring can be produced. So higher number of the recombinant type offspring will give rise to higher recombinant frequency. So higher recombinant frequency will indicate longer map distance in between the two genes. So with that, these would be all 6 important concepts, the important key concept to prevent the confusion when you are answering the questions that's related to the linkage. So whenever you have you get confused with those questions that's related to linkage, always look back into this. Think about these 6 concepts.